Our next speaker is Faisal Mahmood from Harvard University, and he'll be telling us about the use of generative AI in image analysis.
It's a very big file. Thank you so much for inviting me to speak here today. It's nice to see a lot of familiar faces. So I'll be talking a little bit about some work that my group has been doing on multimodal and generative AI for pathology over the past few years. I'll talk a lot about our recent work, but I just want to start by talking a little bit about some of the previous work that we did that sort of led into some of the generative AI work that we have done. Here are my disclosures. So the problem that we are trying to target is that we try to use pathology whole slide images and try to use these images to essentially get to everything in the red box here. So we want to aim for early diagnosis, prognosis, prediction of response to treatment, outcome prediction, patient stratification, integrative biomarker discovery, and so forth. And the problem that we are dealing with is challenging because it's inherently a weakly supervised problem. So we have large whole slide images. There are no annotations available at the level of the slides. Annotations only exist at the at a pixel level. The annotations only exist at the level of the slide that is in the form of pathology reports that can be distilled out based on what interpretation was made by pathologists. So to tackle some of these problems back in 2021, we did some work where we tried to use weakly supervised learning, essentially, architecting attention and multiple instance learning for pathology and making it easy to use pathology whole slide images, slide level labels, while still being able to do this in a data efficient way. The code and everything around this was made publicly available, and it's been used extensively by the community. We then applied this to a number of different problems, including cancers of unknown primary. So when cancer metastasizes, it often becomes unclear where it may have originated. And we posed this as being the weakly supervised classification problem, where we wanted to see if we can use conventional H&E whole slide images to predict where the origin might be. So that's what led to this study. And there are a number of other studies sort of using a similar kind of a technical framework. So if you look at the overview of what we were trying to do, we essentially had some form of feature extraction from these pathology images. So a very large pathology image, it's chopped up into smaller patches. We extract features in some form from these images, and then have some kind of feature aggregation leading towards a prediction. So a lot of computational pathology studies have really focused on these two ends, either trying to improve the feature extraction part or the feature aggregation part, right? So the feature extraction part is perhaps more important, and that's what led us to build, self-supervised models, or now commonly referred to as foundation models for pathology. So what is the role for these large self-supervised models in pathology? So we see them as being generic feature encoders, so essentially meant to replace the feature extraction step in the red box here, which was previously, very commonly done using just a truncated ResNet that's trained on conventional ImageNet features to extract fundamental features from these images, edges, corners, so forth, whatever you can use to describe these pathology images. And we see that these can be applied to downstream tasks. So the real benefit might be that because the feature extractions are richer, now you can use way less data to train these weakly supervised models, and they're not necessarily meant to replace. I think this is sort of a misconception for a lot of people in the pathology community, that they're somehow meant to replace supervised models. That's not true. They're really just very nice feature extractors that are trained on lots of pathology data. So that's what we tried to do with these two studies that were published earlier this year. There's essentially two models. There's the self-supervised model for pathology images only, where we can extract rich feature representations from pathology images, and there's an image text model where we can extract, again, richer image features, hoping that contrasting with text helps improve said feature extraction. So the first model was UNI. It was trained on 100,000 whole slide images that we very carefully selected from the Brigham and Women's Hospital and from the Mass General Hospital, where the distribution was maximized for diversity. So our goal was to see, you know, can we have representation of as many oncotree codes as possible in the data set, and really to focus on getting more and clean data rather than just getting you know, more data that was, for example, just used for screening. So one issue that we have seen consistently, that if you just use, you know, throw all the pathology data that you have at this, it doesn't often converge to a very good model, and that's because diversity matters quite a lot, and the more diverse data that you have, the better kind of model that you're trying to you're going to get. In this particular case, we just used the DynaV2 framework from Meta. It was trained on 32 A100 GPUs, and it was compared with a number of other recently published self-supervised models. And that's what we tried to do here. We basically tested this on region-level classification, region-level retrieval and search, region-level segmentation, and whole slide-level classification for a number of different tasks. And the radar plot is a really, you know, bad way to present this, but it does capture all the all these tasks in like a single image or a single representation. These bar plots really show that what the performance gains were for a lot of these slide-level tasks. I mean, it has, you know, all the commonly used benchmarks in computational pathology, ranging from the Chameleon 16 challenge, which was breast cancer metastases detection, to renal cell carcinoma subtyping, breast cancer subtyping, and towards, you know, sort of more challenging tasks like coarse grain, brain tumor classification, and so forth. So having a pathology-specific self-supervised model did improve performance, but more interestingly, and perhaps for more clinically relevant application, few-shot classification was much better, which meant that we could use very few labels to train some of these models and still achieve reasonable performance, which obviously is helpful for rare diseases, clinical trials, situations where you just would not have enough data available to train some of these models end to end, or they would just not perform as well when using a generic ImageNet trained encoder. We also wanted to see how far can we push the limit in doing this. So we also trained 108 classification problems as just one classifier that can classify 108 different oncotree codes, and what the difference would be from a ResNet-based classifier, and we get about a 12% improvement, which clearly shows that having a pathology-specific encoder makes a lot of sense. In parallel, we did some work on contrasting pathology images with text. So in this case, we used 1.1 million image text captions. So this came from the PubMed Open Access database. It was limited to just pathology slides for human tissue in the PubMed Open Access database, and we used the COCA framework to train this, to try to see if contrasting with text helps improve image representations, which intuitively makes a lot of sense, because this is how humans sort of learn about pathology. Pathology trainees are learning from language cues besides just the images. So it's a distribution for where all the data essentially came from. And we found that the performance was quite good for zero-shot classification, zero-shot segmentation, and so forth. And the goal for doing some of these zero-shot tasks is not to get to clinically usable performance, but to really see how rich the feature representations are, so they can then be used for other, you know, downstream supervised tasks, or just for few-shot learning and so forth. So some more results on zero-shot segmentations. There's a lot more detail and quantitative analysis around these in the publication. We also investigated how it would work on few-shot tasks when using very few labels and so forth. So these models have extensively been downloaded. We've made them publicly available. So we have over 400,000 downloads over Hugging Face. We didn't even know that many people were interested in using pathology-specific self-supervised encoders, but it's very exciting to see how the community is essentially using them. More recently, we have scaled some of these efforts to get to a slide level encoder, where we use the embeddings from UNI, and then scaled it to a whole slide level embedding, where we get a single feature representation corresponding a whole slide image. And then we also contrast it with some of the generative AI work we have done that I'll talk about in a minute, using path chat captions, as well as captions that come directly from a pathology report, hoping that we will get richer whole slide level image representation by contrasting with text. So just some initial results on this. So for a host of different tasks, including for morphologic subtyping, staging and grading, IHC quantification, as well as just looking at how the models scale in terms of data when we have more diverse data, as well as when we have data that's coming from a variety of different sources, how it does on easier problems, difficult problems, and overall we find that models that are trained on more diverse data and models that are contrasted with another modality, in this case text, do much better overall. So speaking of contrasting, we can also think of contrasting with other modalities that have would represent the same data in a different way. For example, transcriptomics, and that's what we did in this study. It was just published earlier this year in CDPR, where we showed that you can get to a whole slide level representation using UNI features at the patch level, and then contrasting with transcriptomics. Initially, this was just done on the TCGA for very specific disease models, and we showed that you could get much better few-shot performance when training in this manner by contrasting with transcriptomics. So this means that if we contrast with molecular information in general, we should converge to a better image feature encoder. So that's what we tried to do here. This is a newer study that's currently in review. We're calling it threads. So we used all the molecular data that was available to us, including from the Brigham and MGH, so transcriptomic data as well as NGS data, and tried to see if we can build a single model by contrasting these image and molecular data sets to eventually converge to a model that would get better image-based performance, and that's what we tried to achieve. And one key result here is that we see that it does particularly well on some of the more challenging tasks, like molecular subtyping, as well as for treatment response prediction, in comparison to models that are trained just on images alone. So this is sort of ongoing work, and we're continuing to investigate this, and we're continuing to see how we can include in more additional assays that might be different from each other, besides the NGS and transcriptomics-based assays that we currently have at the Brigham and MGH. So it's a very interesting direction for us. And we're also trying to see if we can contrast with image, text, and molecular information all together. More recently, we've also been interested in contrasting with spatial transcriptomic data. So we built this data set out of lots of publicly available data, as well as from internal data, where we have histology images and corresponding spatial transcriptomics. We've made all of this data, aligned data publicly available together with the library, how you can work with pathology images and corresponding ST information for a host of different downstream tasks. So this is a direction that we're obviously very, very interested in, because if text-based explanation contrasts well with histology to lead better histology-based image representations, and bulk transcriptomics is doing that, or NGS is doing that, spatial transcriptomics should be sort of very ideal in getting to really high-quality image-based representations when you contrast with spatial transcriptomic data. So the next thing I want to talk about is generative AI for pathology. So my group is in general very, very interested in building a generic chatbot for pathology. So having a single multimodal large language model that would cater to all of human pathology, and why we think that's possible. So if OpenAI can attempt to build a single multimodal large language model that caters to essentially the world's information, pathology is much more specific. So we should be able to build a multimodal large language model that can cater to all of human pathology, and it should have less hallucinations and all the other issues that you see with generic multimodal large language models. So how do we get there? We need a large self-supervised model that can extract rich feature representations from pathology images. We need a vision language model that can encode both images and text. And importantly, we need a large instruction data set that uses pathology images, questions, and has appropriate responses. And finally, we of course need robust evaluation. So our philosophy around building this has been that pathology images are obviously hierarchical. They harbor information at many levels. So we want to have image text representations at all of these levels, but pathology reports don't necessarily have that information. So pathology reports have information at the level of the whole slide. So the philosophy is that fine-grained understanding of pathology origins at cellular level leads to slide level and patient level descriptions, which makes a lot of sense. But pathology reports have images, has information or information corresponding diagnosis just at the level of the slide. So the challenge here is that we needed to collect lots of data at all of these all of these levels. So the user can ask a question corresponding any level, and the chatbot should be able to give a response. So that's what we tried to do here. In the original study that was just published a couple of months ago, we used about 1 million instructions. This included image, text, images, corresponding questions, and responses. A lot of those came from training materials that were built over time at the Brigham and MGH. We also did a lot of manual curation, in particular for having some guardrails built in there for some morphologic descriptions and so forth that we had pathologists collect over time. So essentially had like an image-based feature extractor and a vision language model, and then finally it was fine-tuned with the instructions that we curated. So that gets to the chatbot that we finally were able to build, where we have like a interactive framework where you can look at the whole slide image and ask questions either corresponding the entire image or for regions within the image. For example, here the user is asking what type of tumor this is, and the model would corresponding give like a corresponding answer. Here the user is asking that is this region also from the tumor, and the model would give a corresponding answer. So any questions that the user might have about the image, the user can just go ahead and point to those regions and start asking those questions. So it's a very effective training tool. If there are no questions about the image, you could just ask the model to write a pathology report directly. So the hope is that something like this would be running in the background on its own, and before a user starts using this tool, the report would have already been generated. If any ancillary tests needed to be ordered, would have already been ordered. And I'll give an example of that in a second. Of course, the more context you feed it, the better the report generation capabilities would be, and we have tested that significantly. And another use case is, of course, in lower source settings, and we're very interested in this. So this is obviously it aims to be a single multimodal large language model that can harbor all information corresponding human pathology. And if it can do that, it could be extremely useful in lower source settings where it's very common for medical professionals to take images corresponding pathology slides and send them to their colleagues in more resource-rich settings. And it's often done by coupling a camera to a microscope or a cell phone to a microscope. So if this is the if it would be possible to get responses corresponding these images immediately, it could have a lot of value. So that's what we try to do here and assess whether the model is adaptable to these situations. Another interesting use case is just a camera coupled to a microscope. So only about 4% of pathology in the US is digital. So only about 4% of all pathology slides are digitized and examined on or a monitor. The standard of care remains using a whole slide image, using a glass slide under a microscope. And that's what we try to show here, that PathChat can be used with just microscopes coupled to cameras, which is very common. So in this particular case, the user is asking that this is a metastatic case, and PathChat says this is likely melanoma, and then the user can ask that, what ancillary tests, IHCs, can I use to confirm this diagnosis, and it would give recommendations. At which point, once those IHCs are done, you can come up with that particular slide and feed it into the same context, and then continue to ask questions within the same context. So the context window in the original study was about 4,000 tokens, and we are continuing to try to enhance this and improve this, with the hope that eventually, for every case, we would have the multimodal large language model assess the case, order any ancillary tests that would need to be ordered, analyze those ancillary tests within the same context, and potentially generate a report before the pathologist starts looking at it. So all of this, these demos are great, but we also need to assess the quantitative capabilities of this model. So initially we had a sort of a smaller set that was used to assess this. We had about 260 questions that went through. We pass them through PathChat as well as some of the other multimodal large language models to see, does a pathology-specific MLLM work better? And overall it does. but I think a more interesting assessment was when we asked a panel of 7 board-certified pathologists to rank answers from PathChat as well as from other multimodal large language models, and we found that PathChat overwhelmingly does better, in particular for diagnosis and for microscopy related questions. There are a lot more quantitative assessments around this in the publication. The last thing I just really want to touch upon before we close is transitioning from 2D to 3D pathology and building computational models for this. This is something that we are really interested in. We're collaborating with Jonathan Liu, who spoke yesterday around this. So there are a number of different techniques to image tissue in 3D, and we feel that the real reason why clinical adoption is lacking is because there are not good enough computational tools to essentially point to regions that might need to be looked at by a pathologist, because it will be very difficult for a pathologist to essentially assess entire 3D volumes within a reasonable time frame. To sort of enable this, we built a weakly supervised pipeline, which is very similar to the CLAM-based pipeline that's been extensively used by the 2D computational pathology community, where we have large tissue volumes, we extract regions from these volumes, have 3D voxels or 3D patches, and extract feature representations from those patches, and then lead to an aggregation. Of course, this is everything here is done in 3D, so it's much more computationally expensive. But overall, our results showed for this prostate cancer cohort that both for an open top light sheet microscopy setup, as well as the microcomputed tomography setup, and we used more of the tissue volume, we were better able to separate patients into distinct risk groups. And of course, then once this is done, then you can go in and look at these heat maps which are in 3 dimensions, and then pointing to regions that the model might be using and making its assessments. So I'll stop here in the interest of time, and I would like to thank all the postdocs and PhD students working in the lab, as well as all the funding that we received to do some of this work. Thank you.
Thank you very much. Do you have any questions from the audience? Okay. Candace, do we have anything on the Zoom?
Obviously awesome. Why we wanted to come out and get us, we were excited to see these series of studies come out. One of the things I wanna push you on a little bit is where does it break? Where do you see this not working, or where where we need new training, we need new approaches still to get better classifications or quantifications in a path setting?
Yeah. So I assume you're talking about the multimodal large language model, right? So I think that it gets better with training data, and we're trying to add more and more training data, because human pathology information is finite. So we have the opportunity to, curate very high quality data that caters to everything in human pathology, but we're not quite there yet. So we're about 80% we have data corresponding about 80% of known diagnoses in human pathology. And of course, there are rare conditions, unusual situations. We're lucky to be at an institution where there are 14 hospitals, lots of pathology departments, and lots of data, and we've made good use of a lot of that data. But as I mentioned, that it's very difficult to use just pathology reports and corresponding images for that purpose. It requires a lot of manual data curation for each individual case. One thing that we've recently started to do is to map out the trajectory of diagnosis for a lot of these cases, that you have an initial image coming in, then you order ancillary tests, more images, and then look at the entire trajectory of a diagnostic case, and can PathChat help with those, and what kind of training data can we give it to improve on that. So the areas where it sort of is limited right now, it does hallucinate, it does make errors in morphologic descriptions, but adding more data will hopefully help with those.
I have a question. Great work. Big fan. One of the questions I want to ask is that we have so many very well detailed documented 2D pathologies, but with the 3D pathology that you have shown in the study, which is like probably 30 to 40, how can you leverage what you have in the 2D slices to generatively infer or sort of interpolate in the 3D space computationally?
Right. So I think the interpolation can happen in the 3D space while still interpreting it as if it were 2D slices, right? I don't think we have good data corresponding 3-dimensional interpretation. So if you want to add that additional dimension in the interpretation, there's no existing data that does that really well. This is why in our study with John, we used overall outcomes or overall survival and so forth to train these models rather than using diagnoses that might exist in a pathology report. So we don't have good three-dimensional morphologic descriptions. One idea is that we would get a pathologist to morphologically describe. I mean, they can narrate and it can be transcribed, and then we can contrast with that text. We're looking into that.
Thank you.
Hi, great work. Thank you. Regarding your feature extractor foundation models, can you discuss the advantages and disadvantages, or maybe just your opinions on using these pan-tissue feature extractors rather than a tissue-specific feature extractor foundation model?
Yeah. So we have overwhelmingly found that pan-tissue models work better than disease-specific models, because a lot of and this is just my hypothesis, but because we don't know, but a lot of the fundamental features are common across tissue types. So for the UNI model, initially it was limited to cancer only. Over time, we included cancer, neoplastic, infectious, inflammatory, everything in there, and the model did significantly better. But if you just have, for example, lung tissue and have lots and lots of images corresponding lung, it does reasonably well, but not as good as when you have more diversity of data. And this is in line with a lot of what people are finding in the conventional computer vision community as well. So if you have a feature extractor that's trained on just recognizing cats, first of all, I mean, if that's the case, you probably don't need to have like a self-supervised model. You could just train like a supervised model to do that. But the diversity really helps with fundamental feature extraction.
Thanks.
Thank you. It's really inspiring. And with the recent success in computational pathology has been driven by foundation model and multiple instance learning. And since the foundation model now reach 3 million whole slide image by virtual, and MLL is basically based on attention model. So with these advancements, so what would be the next frontier in computational pathology?
Right. So I think that foundation models are beginning to saturate. That's true. But I think the saturation is sort of driven by how diverse the data is rather than the quantity of data. So there are several models that we have seen which use a lot of screening data, right? So lots of breast tissue, lots of prostate tissue. The slides that are typically created for screening purposes, right? So there's a lot of redundancy across that. And the reason we see them these models perform very similar to models that are trained on just a few 100,000 slides is because the diversity matters, right? So I think we haven't seen an attempt so far where we have maximized for diversity across tissue types, right? And now more recent models are also including like IHC, and they're showing that by including IHC now it does really well on IHC quantification too. So I think there'll continue to be more work on developing better foundation models. But yeah, I think that in terms of the field in general, the generative AI piece is like the missing piece right now, and we'll see a lot of work coming out in that direction. The foundation models are beginning to saturate. There's a saturation in terms of what kind of aggregation functions can you use. So yeah, it's getting closer to the problem of if you have whole slides and labels, you can just train a really good model. So we'll essentially solve that problem with the community.
Thank you.
Hi, so you spoke a lot about how these models can be used for, let's say, downstream endpoint predictions, whether that's response to treatment or diagnosis, et cetera. So I was wondering if you can maybe talk a bit about how can we use AI for pathology models for more discovery as well? Does that look like just looking at attention maps and trying to find new features, or are there other ways to use them for discovering new knowledge?
Yeah. So my thinking around this is that eventually what would happen is that every slide that is ever scanned would get to a feature representation. Either it's at the level of the whole slide or regions within the slide. And as you're collecting more and more data, you'll be able to see what's common across patients and correlate it with some kind of outcome. So if you look at how new disease entities in pathology have been discovered throughout the history, is that someone goes in, looks at lots of cases, finds a new morphologic feature or new feature in general, and then correlates it with the outcome and says, well, this is a new subtype, this is a new And I think what you're asking is that how can that happen computationally? So I think that's we're getting there. A key barrier to that is that a majority of hospitals around the country are still not digital. So the standard of care remains glass slides under a microscope. So when all data is continuously digitized, everything is encoding into meaningful representations. It's much easier to find what's common across patients. How does it correlate with outcome, and hence discovery.
Okay, that's all the time we have for questions for the session. Let's thank the speaker. Thank you. Our next speaker is Edward Schrom from the Germain lab at NIH, telling us about spatial organization of cellular ensembles. Edward? Oh, Zoom.
Yes, thank you so much for allowing me to present remotely to you all from the East Coast. Travel arrangements turned out to be impossible, so I really appreciate the opportunity. Can everyone hear and see everything all right? Is that a yes? Everyone can hear and see?
Sorry, you're not in full screen mode, but otherwise it looks great.
Oh, all right, let's do that. All right, how about now?
Yeah, good. Looks good. Thanks.
Fantastic. All right, well, thank you again, everyone, for allowing me to present remotely. Today, I'd like to show you a computing tool called Spatial Patterning Analysis of Cellular Ensembles, or SPACE, which I believe solves several challenges in the analysis of high-plex spatial data sets. SPACE has been a team effort, particularly among those you see pictured on the slide, and I particularly want to highlight Aaron and Hiroshi, who both gave fantastic talks yesterday morning. Because I don't have to convince anyone in the audience that spatial biology is important, I can jump right into some of the challenges that we face in analyzing high-plex spatial data. So first, we often want to interpret different molecular signals in different ways, which necessitates different pre-processing steps. Some molecules help define and categorize individual cells into discrete types, which can either be left in an image format or could be summarized into tables of centroid coordinates. Other molecules defy segmentation and are better off represented as pixel masks of irregularly shaped, but still discretely categorized objects. And still other molecules are interpreted as continuous expression levels. These latter scalars can be interpreted alone or according to which cell types are expressing them. So ideally, a spatial analysis tool could include all of these representations in a single analysis. A second challenge is that although many great analysis tools already exist, most of them fall into just two camps. The first camp examines cellular abundance in continuous detail, but only in pairs. So whether you use counts of neighboring cells or abundance correlations or shortest distances, these approaches all tell you whether each pair of cell types are closer together or farther apart than expected by chance across the entire tissue. And as a result, we don't learn directly about patterns that involve 3 or more cell types or patterns that differ qualitatively across different tissue regions. The second camp examines microenvironments. So whether these are defined by machine learning algorithms or Bayesian latent Dirichlet allocation or complex neural networks, the end result is the average cell type composition of each microenvironment. Now, this incorporates all cell types at once, but its fundamentally discrete nature is not compatible with continuous patterning like gradients or structural orientations. So each of these broad methods is biased towards the detection of specific types of patterns, and even together, they don't quite encompass the full spectrum of complex patterns we might be interested in. A third challenge is that many of the tools that exist already find patterns that differ from random assortment within a single tissue specimen. But this is not usually our biggest question. Instead, we often really care about how spatial arrangements differ among groups of specimens, like genotypes, treatments, time points, etc. For example, these are segmented images of tuberculosis granulomas, and the primary feature that differs from random assortment in each of them is probably the orange myeloid core surrounded by the cool colored lymphocyte cuff plus some red vasculature. Identifying this pattern in each individual image is important, but it doesn't tell you how your specimen groups differ from one another. So as you might have guessed by this point, I think that my program Space helps address each of these issues. Space begins by capturing a census of each image, which is much like other approaches, except that multiple representations of the tissue can be overlaid and analyzed together. A census covers the image with randomly placed circular neighborhoods and simply counts what's inside each, whether those are cell types, structures, quantitative expression, etc. Neighborhood size can vary, and it should reflect the length scale of the patterns of interest, and the number of neighborhoods total is dictated by tissue size. Within a single specimen, the patterning formed by any specific combination of spatial variables is calculated with a metric that we call cis-mutual information, which is essentially the mutual or the interaction information for the variable's co-occurrence distribution. So for example, for cell types 1 and 2, cis-MI quantifies the structure of those variables' joint distribution that's not already explained by their individual abundance distributions. This calculation is repeated on randomized censuses, which are generated by scrambling the contents of the individual neighborhoods to generate a null distribution of cis-MI against which we can compare the true score and assess significance. Importantly, cis-MI, because of its information theoretic foundation, can discover any arbitrarily complex non-random pattern, not just linear correlations, and it can address any number of variables in an ensemble, not just 2 at a time. So in fact, we usually use space to explore every possible combination of up to 3 or 4 variables at once. Then for any particular ensemble of interest, we next ask, what does that non-random patterning look like? On the joint distribution for the ensemble, we use a self-organizing map to draw a smooth curve through the areas of highest data density and project that curve onto a 2-dimensional plot where each line represents one of the variables to get a picture of how the variables co-vary together across the image following some unseen but informative path. We can see areas where certain variables co-occur or are mutually exclusive, and we can also see quantitative features like smooth gradients. All of this can be done in reference to either null expectations or to a different specimen to see which features are enriched and which features are depleted in the specimen of interest. And you can even draw gates on this covariation plot to see where the corresponding regions are on the original image. We validated this approach on the mouse lymph node, where we already know quite a bit about ground truth. We used a 56-plex IBEX image to segment and define 19 different generic cell types. And then we used space to examine every ensemble, including up to 3 at a time. The top ensembles are shown in the x-axis, the p-value of cis-MI on the y, and the size of the point gives you the magnitude of the cis-MI score. Here, space finds basic patterns, but it also uncovers subtle new details. For example, the most significant ensemble by far is comprised of the dark zone and the light zone B cells, which are tightly clustered in these germinal centers. Accordingly, the covariation plot shows that light zone and dark zone B cells occur in nearly the same place, occupying about 10% of the total image area. And this is enriched relative to null expectations. Additionally, these cells are also patterned not only with respect to one another, but also with respect to the CD4 T cells. So if we zoom in on that covariation plot, we see that the light zone and dark zone actually separate into distinct regions, where the dark zone overlaps with CD4 T cells more. And if we look back at the original image, we can see indeed in all 3 germinal centers, the dark zone is oriented towards the T cortex, whereas the light zone is oriented away. CD4, CD8 T cells and follicular B cells also form an ensemble that had particularly high cis-MI, and their covariation reveals the distinct B follicles and T cortex, which we know to expect. Although these T cells co-occur globally, Within the cortex specifically, we see that the CD4s and the CD8s form an opposing gradient, with the CD4s skewed towards the B follicles and the CD8s skewed away. If you zoom in on the original image to the TV border, you can actually see this gradient in action. Additionally, type 1 dendritic cells peak in abundance close to the CD8s, which are their preferred partner, whereas type 2 dendritic cells peak in abundance close to the CD4s, which are their preferred partner. This entire gradient structure was discovered in the Germain lab in 2019 using a long series of, frankly, quite laborious analyses, and now Space independently discovers it with just a couple of commands. Importantly, these are continuous and context-dependent patterns, gradients, and orientations that are exactly the sort of blind spot patterns that most existing methods aren't really designed to detect. Although I don't have time to show you today, we've also validated Space on a variety of other data types, including 3D imaging volumes, as well as spatial transcriptomics. Space finds patterns not only within the single specimens, but also across groups of specimens, this time using a metric that we call transmutual information. In this case, for any ensemble of spatial variables, we plot the co-occurrence distribution for 2 different specimens. And then trans-MI is essentially the callback-Leibler divergence, which quantifies how different these 2 distributions are. Repeating this for every pair of images, we get a fully connected network where the nodes represent images and the edges are weighted according to trans-MI, such that images with more similar patterning are tightly connected and images with more dissimilar patterning are loosely connected. The modularity of the groups on that network then tells you how systematically this particular ensemble's patterning differs across the groups. Permuting the edge weights gives a null distribution of modularities to again calculate significance. And again, we usually repeat this over many ensembles to discover the specific patterns that differ most strongly across your sample groups. We took this approach on a data set of 30 human tuberculosis granulomas, which was originally analyzed and published by Aaron McCaffrey and our colleagues at Stanford at the time. This data set includes 15 patients of varying severity, from diagnostic biopsies to therapeutic resections, to a few who unfortunately passed away from the disease. Each field of view was transformed into a map of 20 cell types, plus the quantitative expression of 9 different functional markers. And while the original paper, which I highly recommend, focuses on the commonalities of granuloma structure across the patients, Aaron and I used trans-MI to look for differences among the severity groups. And indeed, we found many candidate ensembles. The top hit included the abundance of CD68 macrophages, as well as the PD-L1 expression on CD11BC and 206 positive macrophages, which I'm going to call triple positive macrophages, and IDO expression on neutrophils. What stands out to me most about this pattern is that the triple positive macrophages tend to express PD-L1 in the same areas where the CD68 macrophages are. And in particular, areas with high PD-L1 expression on triple positive macrophages, but small numbers of CD68 macrophages are depleted in the severe cases compared to the biopsy. Whereas areas with moderate PD-L1 expression on the triple positive macrophages, but high numbers of CD68 macrophages are enriched in the severe cases compared to the biopsy. Investigating this further using all of the individual neighborhoods, we found that not only is PD-L1 expression on triple positive macrophages greater in the biopsy samples at large, but the direction of its association with CD68 macrophage abundance is reversed between the 2 groups. So following this arc, but generalizing the axes a little bit, we can imagine a mechanism of disease progression that agrees with the results that Aaron discussed yesterday. Aaron emphasized that in human tuberculosis, granuloma structure is actually associated with immunoregulatory function, like PD-L1 expression. And she showed that CD68 macrophages are associated with bacterial load. So we can imagine a good granuloma with highly immunoregulatory core keeping the bacteria contained. But as CD68 macrophages begin to populate the core, immunoregulatory function like PD-L1 expression starts to go down. And this can lead to a decay of the granuloma structure. As the granuloma structure starts to decay, the CD68 macrophages are free to leave again, but the bacteria can also leak out, potentially leading to severe disease and death. This loss of structure, followed by the total loss of some cell types altogether, is also supported by some cellular diversity metrics that space will also report. So of course, one static pattern really can't prove any dynamic mechanism, but hopefully this shows you how space can take a complex and multifaceted spatial data set and distill it down to the few top candidate patterns that could inspire hypotheses to guide future work. That's all that I have time to demonstrate today, but I encourage you to check out our nearly final manuscript, which is already up on bioRxiv. And of course, space itself is freely available as an R package and a tutorial on GitHub. And you can always reach out to me with your questions. Thank you.
Much. We have time for one question. Yes.
Hi, Dennis here. I was just wondering about the size. So you are kind of sampling, right? When you look into, when you permute the different areas, how does the size of the permutation area plays a role in when you create this kind of trajectory through space?
I'm not quite sure what part you're referring to. Let me
So when you introduce space, the method initially, it comes from exactly here, from like you selecting regions like 1, 2, 3, 4, 5. How big are those regions, and does it matter how big they are?
Absolutely, great question. So what we have found is that the size of these circular neighborhoods ought to be loosely matched to the size of the patterns you're interested in. So if you're interested in cell-cell contacts at the individual cell level, you'll probably want to use neighborhoods that are about 5 to 10 microns in diameter. On the other hand, if you're interested in really broad zonation, you might want to use neighborhoods that are 50 microns in diameter. But the exact size doesn't really seem to matter that much. So if you vary your neighborhood size from 40 to 60 or 70 microns, the results that you end up getting are virtually the same. So you don't need to be particularly precise about that. And of course, you're welcome to use a variety of different neighborhood sizes if you don't know what length scale you're interested in looking at. And something I didn't show here, but that is a feature of space, is you can look at how the results change as a function of the size of these neighborhoods.
Thank you very much, Edward. Let's thank the speaker again. Please take your seats. We are about to begin, and let's welcome our next speaker, David Van Valen from Caltech, talking about foundational models for spatial biology.
Okay. Can folks hear me? Yeah. Okay. Good. I'm going to intermittently be drinking coffee. I flew up from LA, so I've been up since about 3 something. So I need the coffee to stay awake. So today I'm going to be talking about my lab's work on building foundation models for cellular imaging. First, some disclosures. So I am the head scientific founder of a new company, Eisen Therapeutics. They've licensed a number of software tools that are described in this talk. So take everything I say with a grain of salt. And the second is that a number of the results, and actually I'd say like most of them, are not published. So just understand that what I am presenting here today has not actually made it through peer review. This is Dave's word on what these methods do. No one's actually double checked anything. Although we do develop largely openly, so that community evaluation does happen. Okay, so rather than talk about my usual introduction about what's AI, need for data, spatial biology, ... I feel like most people in this crowd are going to be familiar with most of those things. I'm also not going to define a foundation model because there are many different definitions that people use. Some people insist that they have to have a transformer. Some people insist, oh, if it's not doing self-supervised training for representation learning, it's not a foundation model. Practically speaking, for this talk, foundation model is really going to mean AI that works out of the box. And so, rather than having to generate some new data set or generate some new model for whatever it is, whatever problem you're working with, a foundation model should pip install foundation model, run it on your data set, whatever microscope, sample type, day, et cetera, came from, and it should just work. And so operationally, that's kind of the definition of foundation model that's being used. So rather than talk about this stuff, I just like to motivate why foundation models with a one semblance of a scientific story from my lab, and sort of highlighting sort of types of measurements you try to do. You'll start to see like, hey, this is why foundation models could be very useful, and this is how, if we had these things, science could just go much faster at a much different scale, and we'd be able to really phenotype these living systems with the depth that they need. Okay, so in our lab, we spend a lot of time studying cell signaling in macrophages, key innate immune cell type that are largely responsible for sort of sensing the environment in tissues, and then translating whatever it is that they're sensing into some change in cellular and then hence tissue behavior, right? And so they could be sensing endogenous cytokines, immune complexes, et cetera. That information gets sensed by cell surface receptors, gets transmitted down through the signaling pathways, and then eventually leads into some change in cellular behavior, oftentimes highlighted by a change in the transcriptional state of the cell, right? And so it might decide, hey, based on what I've seen, I need to create more cytokines to rally the troops, or I need to push things more towards homeostasis, or I need to remodel the tissue environment to deal with some injury. Skipping many years of hard painstaking experiments done by people who are not myself, it's now appreciated that the signaling pathways that are responsible for doing this transduction actually have dynamic behaviors of activity, right? And so if you look at some of the key nodes, for example, P65 in the NF-kappa B pathway, if you stimulate with one compound, TNF alpha, you'll get one pattern of dynamic activation, oscillation. Stimulate with another, LPS, you'll get a different pattern. And it's to the point where this has been observed across enough signaling pathways where people sort of think there's this dynamic code that's responsible for capturing this information from the environment and making it accessible to the rest of the cell to change its behavior. What's also left out of this slide is that this dynamic activation actually changes considerably from cell to cell. And so if you want to understand, from the information processing perspective, okay, how is this actually happening? And then physically, how is the signal being read out to change its behavior? Your experiments are going to look something like this, right? Where you've got your cell and you've got some measurement of the signaling dynamics. And then ideally in the same cell, you're also measuring the changes in gene expression and then also cell state, right? Cell state loosely captured by cell morphology, organelle organization, and whatnot. Now, this is actually a hard problem. I actually spent, a large part of my early career here at Stanford as a postdoc, creating a first system to be able to measure it. To measure this, what you want to do is, measure the signaling dynamics and then that same cell capture the transcriptome. This is hard because, the two ways in which you measure this, imaging and genomics, typically they haven't talked to each other. But over the past few years, there have been this explosion of spatial methods where you can actually capture that genomic type of information with imaging, right? And so the setup for these experiments then is, you live cell imaging using fluorescent reporters for XYZ signaling pathways, fix your cells at different time points, and then do endpoint spatial transcriptomics to look at the different patterns of gene expression in the individual cells. And there's a lot of technology development that my lab has been doing. A lot of these studies were done in, immortalized cell lines, but what about, the actual human macrophages? It turns out these measurements have never been done in primary human macrophages because it's really hard. They freak out whenever you try to insert foreign genetic material. And so there's a lot of work that went into actually being able to do measurements like this. And so the end result are of 5 years of very hard work done by spearheaded by graduate student Emily Laubscher in my lab, is that we're able to generate these paired data sets where on one hand you see the dynamic signaling behavior, and then on the other hand you actually see the resulting changes in gene expression that happened. And so you could actually go from condition, what the cell is thinking signaling wise, and what the ultimate change in transcriptional state is. And you can do this in a way where it's paired, right? Where for the same cell you can actually capture both axes of information, and this is stuff that we're doing now. You can also add in a cell painting type assay on top of this, so that you get information about cellular morphology and organization. Now, it would be great if I could say 5 years of like hard painstaking labor. Let's just stop here. We got these great figures, just type them up, send it to science, we'll whip out the champagne and celebrate. But it turns out that, this raw data is actually useless for that effort, because this isn't what we want. What we want is the interpretable version of these data, right? The movies themselves aren't of value. It's a single cell signaling traces from those movies that we care about. Getting a series of images of single molecule spots isn't what we care about. What we want are where are the spatial locations of each transcript, the genes identified, and which cells do those belong to, and which cellular record does that belong to. And so it's really, that's a value, right? Where you get these integrated single cell records where, you've got, the signaling phenotype, you've got the morphology phenotype, you've got the gene expression phenotype, everything with single cell resolution, all in the same, all of this information captured about the same cell. And doing that is really hard, and it's really a computational problem. You can start to think about, okay, what are the different things I have to do to get the raw data into this format? And it starts becoming like a very long laundry list, right? I need to have cell segmentation, but it's not enough that I have cell segmentation for my one microscope. Now I've got 2 microscopes. Luxury of being at Caltech and having colleagues like Long Kai who've been able to help us get this stuff up and running. Cell segmentation has to work no matter what instrument we're using. We're doing live cell imaging. That means you need to be able to not just segment the cells, but also track them. You're doing macrophages, and the macrophages will do weird things like eat each other after stimulation. The tracking has to work in that setting. We're doing multiplexed RNA fish. That means for each round of imaging, we need to have the spots detected. But that's not enough. We have to know, okay, if this spot appeared in rounds 1, 3, 5, and 7, that corresponds to TNF as opposed to IL-6. We have a cell morphology component, right? So there's cell phenotyping that has to happen on top of this. And for each one of these challenges, there's sort of a need for a quote unquote foundation model that can just do this on a way that doesn't care about sample type, microscope and whatnot, right? And so, microscope agnostic is really something that makes this type of experiment viable. Sample agnostic, preserves the graduate student's sanity, because, Professor Van Valen might come in one day and be like, hey, I know we've been doing this for, macrophages in a dish, but I heard this, I saw this great talk about tissue slice cultures, can we do the same thing there, right? And rather than having to, stand up a new experimental platform and a new analysis platform, you can literally cut the research effort in half if you have these collections of generalized foundation models. And then easy to use is another requirement, right? Like, wouldn't it be great if the cell culture biologists and the tissue biologists and the microbiologists and everybody could just use the same collections of models because they just worked on everything. Okay, so that's the setup. I don't know if I took up only took up half the time talking about it. I'll spend the rest of the time talking about sort of the different foundation models my lab has either made or is in the process of making across a variety of different problems. Very first one, single cell analysis images for live cell imaging. This is segmentation and tracking. For tissue images, this is whole cell and nuclear segmentation. Looking at the data, you can sort of see the problems in each domain. Tissue images, big problem with cell morphology, just varies widely across all the different types of images that people are imaging. And then there's also differences across the different imaging platforms as well. For the live cell imaging data, the big problem is time. Not only do the segmentations have to be temporally consistent, but there are these dynamic events like cell death, cell division, cells eating each other, that you also need to be able to identify, label, identify as well. Data is, sort of the big unspoken problem, elephant in the room for foundation models in this space. It's not like we can do, what OpenAI has done or Anthropic has done, basically steal the sum total of the internet, train our models on it, kick the IP can, the copyright can down the road and say, hey, we've got, foundation models, give us bags of money, right? We can't do that. It's not because we wouldn't want to. It's not because we have moral scruples. It's just that the data and the labels don't exist, right? And so we've actually been on this effort to create the foundation of data and labels that these vision-centered foundation models actually need. So the core concept is this human-in-the-loop labeling, where most labels are not created by scratch, rather by correcting the mistakes of early versions of models. And to enable this, we've built this software tool, several years old now, Deep Cell Label, browser-native software for doing collaborative labeling of large image collections. And it's capable of now labeling a wide variety of different types of images for a wide variety of different tasks, right? So segmenting cells, tracking cells, identifying cell types, phenotyping positive markers, you name it, odds are Deep Cell Label is able to do it, and do it in a way that's amenable to either experts doing the labeling or crowdsourced workers doing the labeling. On the data side, we've created now a benchmark data set for live cell imaging, where we've taken movies where cell nuclei have been labeled and then imaged over time, and then created single cell labels, so everything has been segmented, tracked, cell divisions identified. This data set, I think we're calling it LiveCellNet, contains over 500,000 single cell labels spread across about 15,000 unique trajectories. Not surprising, the hardest part of this problem is just identifying the cell division events. And most existing data sets of this kind do not have enough cell divisions labeled to enable models to do this accurately. With a better data set, the accuracy of the cell division detection goes up substantially. Similar efforts have been ongoing for tissue images. Noah Greenwald worked with our lab for several years building out a data set called TissueNet, which was published now a few years ago, which basically created labels for over 1 million individual cells, both paired nuclear and whole cells, across a wide variety of different images, tissue types, and disease states. And with that data set, we built this method called Mesmer, which does both nuclear and whole cell segmentation. And because it was trained on this large, diverse data set, it works well across these different domains. Okay, so that's great, a lot of progress. But the challenge, the issue that this field has faced over the past couple of years as these data system models came online is just fragmentation. Is that every different domain would have its own set of models that worked well for that domain. If you have tissue images, then you had Mesmer. If you had bacterial images, then you might use Omnipose. If you had cells and cell culture, you might use something else. And for this wonderful foundation model worldview, ideally what you'd have is a single model that's able to do everything. And so we've been working on, towards building such a model over the past couple of years. The effort has led to this model called CellSam, where we've sort of taken the sum total of all of the labeled cell segmentation data, and then adapted a method called Segment Anything model from the folks at Meta, that has proven very flexible for producing accurate cell mass for a wide variety of images. The way SAM works is, you sort of take an input image, and then you can ask it to generate a mask for an individual object in that image by providing a prompt, right? So this prompt can be a point, a bounding box, even a crummy mask that you have available. And based on that prompt, it'll generate one mask for that one object. This is very powerful for doing human-in-the-loop labeling, because this allows for interactive labeling modes. The problem is that there's no way to automatically prompt SAM for every object in the image. And so what we've done is we build an additional model called Cell Finder that automatically generates bounding boxes for everything in the image, and those bounding boxes are then go to prompt a fine-tuned SAM model to generate segmentations. This is work that we're in the midst of getting published. But the short version is that SAM works better than anything else we've seen across all the different domains that we've tested it on. And it's to the point where it's basically supplanted all of the segmentation methods that we use internally in our lab. And we generally have taken a kitchen sink approach for these labeling projects. Right now, most people in our lab are using SAM. And what's really nice is that it just works across these different domains, even beyond them. So last week, a colleague of mine, Diane Newman, came in with some EM data. Dave, can you do something? Tried SAM, and with a little bit of fiddling, we ended up getting quite good results. Okay, remaining foundation models are for problems that happen after cell segmentation, and so this is cell phenotyping for spatial proteomics. What are the cell types? What are the positive markers? And then spot phenotyping for spatial transcriptomics. For cell phenotyping, the reason why this is hard, some of it is due to just the artifacts that are present in these data. You get segmentation errors, you get labeling noise, you get spillover of markers between neighboring cells. This can be solved with good labeled data and vision models. The big problem is generalization, and that is just alluding to the fact that there are a wide variety of different spatial proteomics platforms that are now available. Some of them use imaging with light, others use imaging with mass spectrometry. This again can be solved with labeled data and vision models. But the bigger problem is just that there's just a wide variety of things that are being imaged, and the marker panels differ drastically across these different experiments. And so for about a couple of years, we had a technician in our lab, Laura Prodhan, and what her sole job was just to go find all of the publicly available spatial proteomics data, ingest it, and get it to a form that was sort of machine learning ready. As a result of this effort, I think we have, on the order of 20 or so data sets. We've ingested all of the available cell type labels, after passing through some quality control, and also generated a fair number of labels ourselves. And across all these data, there's about 31 cell types that are represented, but then also 198 different markers that were present in all of these experiments. And so this is, at least in my view, a big challenge for any model that's going to be doing generalized cell phenotyping. And there are some things that you need for generalization, normalization, access to the full information content of the images. But I think the reality is that, if you have, nearly 200 different markers, and this is something that's, just growing, it's going to get larger, not smaller, then really what you need are AI models that are just aware of the biological information in the marker panel itself, right? And so it knows, what CD4 is, it knows what CD8 is, it knows what CD45 is, it knows what a T cell is, it knows what a B cell is, and it can use the semantic understanding to expand the space of markers that it's capable of doing, and cell types it's capable of doing cell phenotyping for. And so, as I alluded to, you don't get to do these sort of efforts without first, generating these data sets. And so we built a data set for generalized cell phenotyping. We're calling it expanded tissue net, over 10 million single cell labels, both segmentations plus cell types, across a wide variety of tissue types, and also a wide number of lineages. This includes publicly available data, as well as data that was made available by the HubMap Consortium. And with this data set, we've been able to develop this model called Deep Cell Types, which is a language-informed vision model for doing cell phenotyping. And roughly the way this works is, you take your images of each stain as well as the name for each stain. The images for each stain get passed through an image encoder. So this is a simple convolutional neural network that creates a vector summary of what was in that image. And then the markers get passed through a language encoder to create a vector summary of what that marker is. And so this language encoder is this two-step process. Does this work? Oh, okay, well, yes, but it's quite small. So the first process is we ask a language model, hey, what do you think about this marker? Right, tell us everything that you know about it. You get a couple sentences long summary about a given marker, and then there's a second language model embedder, which takes that summary and then creates a vector summary of what the language model thought. And of course, we curate this to make sure that the language model isn't hallucinating information that's not factually correct about any given marker. We also do this process for the cell types, right? And so we ask, hey, what do you think about, T cells, B cells, tumors, what cell type, what markers might they be expressing? Give me some biological information about them. and you'll end up getting vector summaries for the cell types as well. And so for the image summary and the language summary for the markers, this get passed into a transformer module, which sort of mixes the information, and you kind of view it as, sort of inspired by how humans look at this information, looking across different markers. Does this image look, biologically reasonable or not? And then what you end up getting at the end are sort of these, these vectors that have both the image and the language information. And as well as a class vector, which sort of summarizes everything that's present across the different markers. The attention scores for this class vector and the different markers can then be used for doing the self-phenotyping, the marker positivity prediction, and this class vector can then be used in combination with the cell type embeddings to do the cell type prediction as well. And this actually ends up working quite well. And so here we see some example data taken from expanded TissueNet, held out, of course, where you can see different platforms, IMC, codecs, and whatnot across the different tissue types. The accuracy is fairly good. So you can see the F1 scores for the cell type prediction detection task. And then you can just look at the embedding space, and you'll see like the different cell types actually end up occupying quite distinct locations in this embedding space. Kind of not suggesting, kind of demonstrating that these deep cell types has actually learned a meaningful space about this shared vision and language information. And there's more rigorous benchmarking that we've done too, but that's not as impressive as the pretty pictures. Similar story for the spot phenotyping. Image-based spatial transcriptomics is essentially doing sequential rounds of single molecule RNA fish, and for each round you have to both identify where the spots are and then align the rounds and then identify which detections actually correspond to which genes. One very hard part about this problem is the spot detection, and this is largely a data issue. For these experiments, there's just far too many spots to ever label manually, and then also just the notion of what's a real spot and what's not, something that's a little nebulous. If you could write it down, that would just be your algorithm for doing the spot detection. And so what we've put together is a weakly supervised deep learning approach for doing spot detection and then integrating it into a integrated pipeline for image-based spatial transcriptomics. And roughly the way this works is, we collect representative spot data, fine-tune a collection of manually fine-tune a collection of classical spot detection algorithms, and then we do generative modeling to find consensus among these algorithms. And these consensus labels are what we train our deep learning model on. And we find that when we do that, this generative modeling effectively denoises the labels, and the resulting models are considerably more accurate. And so you can pair that spot detection model with generative modeling for doing the spot decoding, and then look in the grab bag of foundation models and see which ones exist that are paired to the other things you want to do for image-based spatial transcriptomics. It turns out that cell segmentation is the other big thing that you want to do, and so you can use Mesmer, you can use CellSam, any other comparable method, and with the spot detection methodology, you end up with a integrated pipeline for doing image-based spatial transcriptomics, where data comes off the instrument, segment the cells, find the spots, decode them, and then you're able to generate these spatial maps, either in tissues or for the cell culture experiments that I presented. So I'm pretty sure I'm out of time. If not, I've gone over, and so I will stop here. Just give a huge thanks to my lab. The work I present has been ongoing effort for several years. Big thanks to collaborators as well. Mike, who's worked with us for over the years, Wong Kai, Isong Yu, and Yak Ren, who's at the Weizmann. Then also a big thanks to a variety of organizations for funding and support. So that I'll stop talking and open the floor for questions.
Thank you, David.
Really interesting work, David. I had two questions. The first was quick. For a deep cell type,
I might have missed it, but do you combine that with cell segmentation as well, as you alluded to?
Yeah,
with the Polaris?
so that's the precursor step. So everything has to get segmented, and then once you segmented, you crop out fields of view around each cell, and that's the input for deep cell types. Whether that remains the case in the final model, we'll see. Right now, that's generated with Mesmer. We might end up redoing with CellSam. It's a work in progress, but right now, yeah.
Awesome. And then the second was for CellSam. One of the challenges with segmenting cells and tissues, for example, is they're multinucleated sometimes, or smooth muscle cells have these weird shapes. How does it do on that?
So on tissue data set, CellSam outperforms everything that we've tested on. That being said, the way I would approach the problem is just, what's your definition of what's a cell? Right? Is it the whole cell mask or is it the cell nuclei? Is that what you're calling a quote unquote cell? The data structure that'd be matched best to that problem for the multinucleated cells would just be do the whole cell segmentation, then do the nuclear segmentation, and then which nuclei exist in which whole cells, right? And then use the whole cell marker as your marker of what's a cell, but that cell might end up having, that cell in quotation marks, might end up having 5 or 6 different nuclei.
And for weirdly shaped cells,
we only have time for one more quick question. So,
yeah.
Really nice work. I'm wondering for deep cell types, you showed that while you're using data from different technologies, and these technologies are as well at different resolution, I guess.
Yeah.
So do you find that just the more data, the better, or is sometimes having something too different not helping the model to perform better?
So more data isn't necessarily better. So there are things that you can do to standardize, right? So, reshaping things so the pixel size are the same, but more data isn't necessarily better. And I'd say there is a lot of data, but a lot of good, but the amount of good, high quality, clean data is less than one might think. And I'd say the most common failure mode we see with deep cell types right now is just weak staining, where there's some marker which is being stained, but that epitope isn't in sufficient abundance, and the signal that you get is quite weak. And in some cases, weak to the point where the normalization methods that you need to get the generalization to work end up washing away that signal. So like, how long is that going to, and that's more of a data issue, right? And there are things you can do on the measurement side to improve that signal amplification and whatnot. But more data is not going to make that better. If anything, it's going to be better signal, better data, right? And so there's a point, you have to understand there's a point at which, more AI, R&D, more labeling, more is not going to actually solve the problem. And then that's, diminishing returns for the research time and dollars. So you got to put a pause on that and, let the data, sort of meet the algorithms in the middle.
Thank you.
Thank you very much, David. And our next speaker